Good evening, students. This is the chapter number 4, Principle of Inheritance and Variation. Before going to this chapter, I just want to remind you all the three chapters already has been published in my YouTube account. So you have to go through the videos seriously, and also you have to go through the video in the connectively mode. So not spending so much of the time in these types of the morality and something, we are going to further start this chapter. So, what is the principle of inheritance actually? Actually, genetics is the main base of the inheritance, and because of the genetics is actually the study of a principle and mechanism of both heredity and variation. So, heredity is actually the heredity and variation. I'll separately tell it to you. Heredity is actually the character which is transferred from one generation to another generation, that is called as the heredity. And if we will go for the variation, the degree at which the progeny is differ from the parents, how the next generation is different from the parental generation, that is variation. But before that, you have to understood that alleles are the things which are really important to generate this type of the variation. Allele actually are in pairs, and these are the contrasting characters. The next part is a Mendel. Mendel actually the main base of this chapter. Mendel is a scientist who worked on a garden pea for the 7 years from 1856 to 63, and he works on the seven character of a garden pea. First thing is why he choose only pea. He choose the pea because the pea plant, that is Pisum sativum, has a contrasting character. The lifespan is very short, and it is self-pollinated plant, annual plant, whole year. In 3 months give the result and easy to cross. What are the seven character? Actually, the character is divided into two parts. One part is called as a dominance, another part is called as a recessive. So the dominant and recessive part is both different. Now this dominant part, if it is tall, then the recessive part is a dwarf. If the dominant part is yellow, the recessive is a green. The dominant part is a green, the recessive part is a yellow. The dominant part is inflated, the recessive part is constricted. The dominant part is a violet, the recessive part is a white. The dominant part is axial, the recessive part is a terminal. The dominant part is a round, and the recessive part is a wrinkle. What are these dominants and recessive? Actually, the dominant and recessive is the dominant are those type of the species which those type of the character actually which suppress the another character, and the another character which is suppressed is called as a recessive. Now, the first third part is the inheritance of one gene. The inheritance of one gene is actually a type of a monohybrid cross. A type of a monohybrid cross which coming in the first generation, and in the first generation it gives the result. So if I cross the pure tall plant, that is capital T, it is called as a homozygous if the character is same, the alphabet is same. And pure dwarf small t is actually a recessive and homozygous. The cross found in the F1 generation is capital T small t, which is a type of hybrid. It means it is tall actually because the dominant part is a tall, but it is an heterozygous tall. So when we are going for the selfing of capital T small t with the capital T small t, it gives the result of homo and hetero also. So, the homozygous capital T, one part is formed, another part is small t and capital T, third part is capital T, small t, and fourth part is small t, small t. So, the small t only represents the one upon the fourth part. If the four block is there, the one part is of recessive, and the three part is a dominant. Now, if there is one cross that is called as the test cross, or you can say back cross also. Um, Test cross is actually the cross between the F1 hybrid to the recessive parent. So capital T cross with small t, then the result is capital T small t heterozygous dominant is 50%, and small t recessive is also coming 50%. But when we go for the back cross is between the F1 hybrid and a pure tall plant. This pure tall plant means homozygous dominant. It gives a result capital T small t into capital T. The capital T pure homozygous is half, and is capital T small t heterozygous dominant is half, so it's 50-50%, but no chances of recessive. So this is the multi-hybrid inheritance of one gene monohybrid cross. If we go for the Mendel's law, the Mendel gave the law in 1865. First law is law of dominance, another law is law of segregation, and another law is law of independent assortment. So law of dominance is actually the dominant allele suppress the recessive allele, as you already saw before. Capital T dominant crosses with the small t gives a dominant result heterozygous, whether it is heterozygous, but actually it is dominant now. So if it is a dominant, then you can say the dominant suppress the recessive. Now the next one is the law of segregation. The genes free to segregate, that's why it's said by the Corens in 1901 that any type of the gene which is present in the chromosome can easy to segregate and cross with any of the genetic behavior. So capital T is the small t gives capital T small t. Why the hybrid is formed? This hybrid is going to be formed by the progeny. The phenotypic result is 3:1 and the genotypic ratio is 1:2:1. So it created two type of the factor. One factor is called as an incomplete dominance, another factor is called as a codominance. Let me completely tell you. Incomplete dominance is actually no one will become dominant in the next generation. It means the result is something mix. Example 4 o'clock plant, carination plant. Why? Because just like as an example, if I cross two breeds, one is red flower and another is white flower, the result of the next generation is pink flower. It means the phenotype is 1:2:1 and the genotype is also 1:2. Look at it. You have to learn this type of the phenotypic and genotypic ratio. In this type of a cross incomplete dominance, the ratio is always 1:2:1, whether it is phenotype or a genotype. Now go for the codominance. Both show the dominance character. It creates a different type of multiple allelism like type of the term. The one part is ABO blood group type given by Landsteiner and another is by AB blood group type given by Stinney. So ABO blood group, it means O consists of IO that becomes a recessive. The A part is also dominant but actually mixed dominant. If IA is there, I is the value which create type of a factor of a blood group. So AA is dominant. If it is going mixed with the O, then IAIO becomes heterozygous dominant. Go for the B. Actually, the both blood group is A. IAIA is also a blood group. IAIO is also a blood group. The same condition with the B, homo and hetero. And if we go for the AB, as you see here, one is dominant, one is the recessive, and another is a hetero homo. So this type of the allelism is called as a multiple allelism. So here AB is actually written wrong. It is IAIB that becomes a dominant. So the next one is due to some type of the mutation or type of a single gene pleiotropy is there, and the example is phenylketonuria because by this enzyme phenylalanine hydroxylase is not going to be formed there, so it causes a mental retardation. I will tell you in the further. So go for the next one. Inheritance of a 2 gene. The 2 gene is a dihybrid cross. The dihybrid cross is actually the inheritance of 2 gene in which the round and yellow two characters, one is a shape, another is a color, is going to be taken. So round yellow is represented by capital R, Y and Y capital. Now if it is going for the round, the opposite part is wrinkle, recessive mode. This wrinkle consists of a smaller and the small y representing the green. So if we mix, you also have to make the progeny like this. They have to take the round and round there and mix with the both patterns of Y and Y. So you get four type of the ligament progeny. And this ligament progeny is further used in dihybrid cross. So law of independent assortment will say that if we cross the two characters in the same time, it will create a heterozygous type of a character. But showing the dominant, it is also round. See, capital R small r is also round and capital Y small y is also yellow. So if we selfing it, it creates a four type of a progeny you already see here. Four type of the progeny. Wait a second. So the four type of a progeny have you seen here, ry, small y. So if we cross with each other, it creates a phenotypic ratio of 9 ratio 3 ratio 3 ratio 1. Stop, pause the video, see the crosses, and you will easily find out how the cross is going to take in place. Sometimes the genotypic ratio is not going to be asked, but here I will show you the genotypic ratio of this phenomenon also. One more thing, if we go for the trihybrid cross, see the trihybrid cross. This is actually a trihybrid cross. The breeds are find out but it is not in the course of class 12. Don't worry about it. You have to go through it and have to find out that these type of the crosses are going to be formed somehow. Phenotypic ratio is going under 1, 6, 15, 20, 15, 6, 1. Now the next one is limitation. Why the Mendels have some type of the limitation? The one limitation is law of independent assortment is not working on the gene on separate chromosome. The second one, linkage is not confirmed the law of independent assortment. Linkage, I'll tell you now. Crossing over needed and not apply on the X-linked traits type of the diseases. Now chromosomal theory of inheritance. Who given the chromosomal theory of inheritance? Sutton and Bowery gave the experiment. And experimented by T.H. Morgan. Morgan actually working on the fruit fly. So that's why this fruit fly name is Drosophila melangosta. So the genes actually located on a specific loci and segregate and assorted. So this is called as a linkage. Linkage is two type. One is linked gene, another is an unlinked gene. Linked gene in which genes are present on the same chromosome, and unlinked gene in which genes are on the different chromosome. But the linkage is also two type. One is a complete linkage. Complete linkage actually there is no change of the separation, no chance of a separation of genes. They are very closely related to each other, and the crossing over will not separate the genes. But if we go for the incomplete linkage, the full chance of a separation of the genes are going to be there. They are located distantly, and during crossing over, they are going to change. As you see here, the gray and long crosses with the black and vestigial and crosses in F1 generation. The gray long is 50% and the black vestigial is also 50%. But here, what is going to be happen? The black body and purple eyes crosses with the grey body and red eye, and the grey body red eye 47% is the grey body and 47% is the red eye. And here the black body and purple eye is 100%. If we cross with each other, the grey body and red eye and black body purple eye same color is found. But if the crossing over is there, then it will form black eye red eye, black body red eye, and here the gray body purple eye. It means the color and body shape is also going to be exchanged with each other. So somehow the parental generation here is going to be formed, and here the recombination is going to be formed. So you have to see about it. Now in sex determination technique in 1902, it is given by F.B. William Stephen. It is of three type. One is XY type, another is XO type, and third one is ZW type. Let me see. XX and XY type consist of XX is female, XY is a male. It is present in human beings. If we go for the XX and XO type, XX representing the female and X0 consists of male. It is coming in the insect roundworm. And it is ZZ and ZW type. Actually, this is different. The ZZ representing the male and ZW representing the female. It is coming in the birds. So these type of the sex determination is also coming. Somehow, the question is going to be asked how the birds is going to be represented by the chromosome. So the next one is mutation. Actually, it is caused by the mutagen. It is caused by the mutagen. Mutagens is of two type. One is physical and the is chemical. If it is physical, then it comes under the ultraviolet rays, alpha rays, beta rays, gamma rays, X rays. And if we go for the chemical, it going for the mustard gas and nitrous gas. So there are types of the mutation. One type is a point gene mutation. The change in the gene structure alter the information. The structure of a gene is changed, and the information is not correctly transferred. Frame shift mutation is the addition or subtraction of a nitrogenous base, so that's the codon cannot be read by the mRNA and different protein is formed. If we go for the chromosomal purpose, the chromosomal consists of a four type of the things: deletion, duplication, inversion, reciprocal. And if we go for the numerical purpose, it is of three types: haploid, diploid, and triploid. Let's go for the deletion and that one. So deletion actually when the portion or part of a chromosome is lost. Here you see the diagram and one part is lost. So what is duplication? When any portion become double after mutation, like eye size in Drosophila. The two parts is there, the blue become double. Now, inversion. When the sequence due to rotation or folding become change, hence the sequence is changed. One, the pink one is in the front. Now the blue one is in the front, and the last one is reciprocal translocation. It can happen in two chromosomes of homologous pairs which exchange their chromosome. Look at see the upper part, and now you see the down part. The pink color is going to the second one, and the blue pattern is going to the first one. Now, numerical mutation you see here, some type of the number of the chromosomes are fixed. These chromosomes are present in the form of complete set. Example, single set is haploid, two set is diploid, and many set is polyploid. Now, we go for the next one. So the next one is actually due to the chromosome number, there are two types of the ploidy is also found. One is aneuploidy, another is euploidy. If it is aneuploidy, listen it very carefully. If it is aneuploidy, the chromosome number which is not the exact multiple of basic haploid chromosome because it is of two types: monosomy, trisomy. Monosomy means 2n minus 1 is going to be happen there. The number n set, the double of an n set, multiple, exact multiple of that set minus 1. And trisomic means exactly multiple of double is plus 1. Means one chromosome, one gene is extra here. Now in euploidy, here's different thing. The chromosome number is the exact basic copy of haploid chromosome. Like triploidy, 2n plus n becomes 3n, and tetraploidy, two additional sets are formed. So two types of the things are there. Now what are the disorders? Disorder actually called as a Mendalian or chromosomal. The Mendalian, it is of I think six types. Number one is sickle cell anemia. What is that? The male become affected, the female become affected or the carrier, because the sixth gene of hemoglobin where the valine is going to be formed is displaced by glutamine. Now the next one, thalassemia. Here the hemoglobin become abnormal. Anemia can cause. It is of two type: alpha thalassemia and beta thalassemia. Treatment is only one: two, blood transfusion, blood marrow transplant. Now the third one is phenylketonuria. I'll show you how the phenylketonuria work. No enzyme is going to be there in the body to make a tyrosine. So phenylene is not converted into tyrosine, it becomes converted into phenylpyruvic acid. And because of that, the body consists of mentally retarded. The next one is hemophilia. What is hemophilia? Blood clot will not occur. X-linked disorder is there. Actually, this is called as a royal disease because firstly it is happened and find out in Queen Victoria of. You have to comment it where the Queen Victoria is found. Where is Queen Victoria present or was present? Now the next one is color blindness. You see there are different type of the blindness maybe occurs. Somehow the person is not able to see the color vision. It is also techniques which are checked in the army and defense system also. So somehow it is not differentiate between a red and a green color. That is a color blindness. The next one is chromosomal. Chromosomal is Down syndrome. It is of two types, Turner syndrome and Klinefelter. So Turner syndrome is actually female has one X chromosome. The female becomes sterile and mentally retarded, but Klinefelter syndrome is happen with male which has one X extra chromosome. It becomes a sterile and mentally retarded. So these are the disorder it can be asked in the question. Now the next one is pedigree analysis. Actually, pedigree analysis happened because of the record of the generation. It collect the family history, make the history, and make a family tree. It is helpful to find out the genetic problem. Even the genotype should be known. It is helpful to study hereditary. Go for the pedigree analysis chart. It is represented by like this square male, two males, proband male. If it is some type of the problem, then male is dead, deceased. Affected male is black. Affected by history is somehow like this. Examined male is plus the fraternal twins, identical twins, they are combined. Adopted male. Female is also somehow like this. You have to pause the video and write these things in your copy. The third one is this sex unknown, lived one day, stillbirth, miscarriage, pregnancy, marriage, consanguineous marriage, extramarital mating, like this. There are some crosses also. You see here, the cross between a tall red and a dwarf and white. So it is a dihybrid cross. There are ratio also there. You have to pause the video and find out the ratio which are very important: monohybrid cross, phenotypic ratio, genotypic ratio, test cross ratio; dihybrid cross, phenotypic ratio, genotypic ratio, test cross ratio; complementary gene also, dominant epistasis also, recessive epistasis also; incomplete dominance, phenotypic ratio, genotypic; codominance, phenotypic ratio, genotypic; trihybrid, phenotypic and genotypic ratio; type of phenotype 2n, type of gametes 2n, type of genotype 3n, possible gametic number n plus 1 upon 2, polygenic inheritance, a different type of things you have to find out. Now go for the PYQ. So first question is, mention any two contrasting character with respect to the seed in the pea plant that were studied by Mendel. You already know the seven character. On what basis the skin color of the human is considered as a polygen? Because skin color is different time na, somehow Savla, Gora and black also. British scientist R.C. Punnett developed a Punnett square. Mention the possible reason why he performed these type of things. Fourth question is, actually the monohybrid cross in F2 generation ratio is 3:1 and phenotypic it is 1:2 ratio. So why they are different? Fifth is, what is test and back cross? You already know. What is the law of segregation is the sixth question. And the seventh one is state about the linkage and the types. So you have to go through the video seriously, slowly, and very slowly. I will put the things if the PDF is ready then. But before that, you have to go through the video and find out what is the next one. So thank you. Have a nice day. Like, share, and subscribe, and also share with your friends, those who are going for the board examination of the CBSE board. So recently I'm going to expose the questions and answer also here. So don't worry about it. Have a nice day. Okay, thank you.